Okay, so this is the fourth part of section 1.1 in the genetics unit. This will focus on page 14 in the textbook, and it is a relatively short lesson. So this lesson is designed to give you a bit more background information, as protein synthesis can be a bit confusing. Step 2, after transcription, which again is the process of mRNA being made and moving out into the cytoplasm. The second step after that is translation, which occurs in the cytoplasm. So you might remember that ribosomes are responsible for protein synthesis. So this was from grade 8 and a little bit of grade 9, and this is making proteins. The mRNA, which is just a code, is read by the ribosome in order to make a protein. Reading of this code always starts with what is called a start codon. And the code for this particular start codon is a triplet, so 3 bases, and it is AUG on the mRNA. So the purpose of this start codon is so that the ribosome knows where to line up and start reading the code. Every single protein starts with this AUG start codon. So the mRNA code is made up of groups of triplet codes known as codons. Each codon codes for a specific amino acid. So for example, AGC codes for the amino acid serine. UGC codes for the amino acid cysteine. It is good to know that there are a ton of different codons. You are not responsible for memorizing this. We will have a chart provided for you that you can use when answering these sorts of questions. It takes 3 bases to make 1 codon. And why this is because there are about 20 total amino acids, and we only have 4 bases in our alphabet. So the code that we are creating is based on the 4 possible nucleotide bases, so A, C, U, and G. So if we were to just use a single nucleotide, there would only be 4 possible codes, right? So A would be 1, U would be 1, G would be 1, C would be 1. But if we were to pair these nucleotides, so double nucleotides, there would be 16 possible codes, so 4 squared. So this means that we could rearrange these pairs of those 4 possible letters in our alphabet in 16 different ways. So for example, AA, AG, AC, AU, UA, UG, UC, UU, and so on. You could do this for the rest of the possible pairings, and you'd get 16 different possibilities. So that's still not enough for our 20 possible amino acids. So by utilizing a triplet code, we can have more than 16 possible amino acids because there are 20 of them in reality. So by using the triplet, we have 64 possible codes, which is more than enough to cover our 20 amino acids. So essentially, there are 4 possibilities for the first letter, 4 possibilities for the second letter, 4 possibilities for the third letter. So 4 times 4 times 4 is 64, or 4 to the power of 3. So every possible amino acid has at least 1 possible triplet codon. Some of them have more than 1 possible codons. The amino acid associated with a given triplet does not seem to be chemically determined. So the presence of the anticodon on one end of the tRNA, which is transfer RNA, we will talk about that in a minute, does not chemically determine the amino acid attached to the other end. So you can see here there's a triplet code, and then this is referring to the amino acid here. And sorry, I left this out for the 64 part. Okay, so let's figure out how to actually determine our amino acids for a given codon. So the amino acid for each codon is given in the tables below. So these are the tables I was referring to. You will always be given these charts. You will not be asked to memorize, although in university you may have to memorize them if you take a biochemistry course. So let's work on how to actually read these tables. So for example, on the side here, I've got first letter, second letter, and then these over here refer to your third letter. Your first letter could be a U, a C, an A, a G. Your second letter could be a U, a C, an A, a G. And then the third letter could be U, C, A, G, and so on, U, C, A, G, again and again and again. So basically it's just creating that triplet. So if your first letter is a U, if we're starting here, the second letter could be a second U or a C or an A or a G, and same with the third letter. So for example, if we try and use, we're reading a code that is U, U, U. So our first letter is U, our second letter is U, and then the third letter is also a U. We're looking in this top box here in the first row. So that means it's this amino acid here. So that is phenylalanine for the code UUU. So notice with phenylalanine, so again, that's right here. There are two possible triplet codes that could code for that. So UUC is also phenylalanine. So if the third letter is changed from a U to a C, it's still phenylalanine. So this is because remember that there are 64 possible codes and only 20 amino acids. So we will have repeats. So there will be 1 to 4 possible repeats for kind of each amino acid. So whatever your codon is, you're then able to figure out what amino acid is listed using one of these two charts. So the second chart, which a lot of people prefer, is the round chart. So the first circle refers to our first letter in the code. The second circle refers to the second letter in the code, and then your third would refer to the third letter in the code, and you basically work your way out. So again, UUU refers to phenylalanine. This chart works exactly the same as the other one. They'll give you the exact same possible amino acid. It's just basically which chart works best for you. So let's work through the first example here at the bottom. So CAU. For CAU, our first letter is a C. So we know it's going to be in this row. Our second letter is an A. So it's going to be in this column here. And then the third letter is a U. So it's going to be across to here. So this is telling us that it is histidine. If you were using the circular chart, we would start in the center with C. The second letter is A, and then our third letter is a U. So right here, so again, histidine. Okay, so I would like you to try out the remaining possible 5 codon sequences here. So pause here, work through these, and then in addition to that, you are going to work through these 2 questions as well.